Hi, I'm Steve Kinsey, Technology Licensing Officer with the Commercial Ventures and Intellectual Property Group at the University of Maryland, Baltimore. This video will give you a description of a new and innovative approach to computer-aided drug design called Site Identification by Ligand Competitive Saturation, or SILCS for short. You'll hear a description of this technology from the inventor himself, Dr. MacKerell, professor in the School of Pharmacy. Dr. David Weber, professor in the School of Medicine, will discuss some preliminary results using compounds designed with this method. And finally, we'll hear an industry perspective from Dr. Natalie Eddington, Dean of the School of Pharmacy, formerly Assistant Director of New Drug Development at Pfizer.
The SILCS technology is a novel ligand design technology that takes mathematical models and uses them to understand the interaction of ligands and the target molecule, be it a protein or, say, possibly RNA. Up to now, computer-aided ligand design technologies have been somewhat limited in the fact that they're used for one protein, one site on the protein, and typically one ligand on the protein surface at a time. The accuracy of the SILCS technology is based on immersing a protein in a solution of organic molecules. This allows us to then determine the three-dimensional probability distribution of where different functional groups want to lie on the protein surface. The advantage of this is this allows for orders of magnitude increase in the efficiency of the methodology over these previous computer-aided ligand design technologies. This allows for the drug discovery of the ligand discovery company to more rapidly bring a product to market, thereby saving resources, maximizing their income over a longer period of time. The SILCS technology can greatly facilitate the ligand discovery process by saving both time and money. We can literally shave days, weeks, maybe even months off the design process. Here in a ribbon diagram, we show the structure of a protein, but we also add in water molecules. In addition to water, we can have, for example, benzene molecules. In addition to benzene, we can also have propane molecules, which I now show in green. These molecules, as well as the protein, are all flexible, and the molecules are searching the surface of the protein to identify sites on the protein surface to which they like to interact. At the same time, they're competing with each other. So the SILCS technology actually identifies the sites on the surface for which, say, benzene wants to bind more than propane.
Proteins have a million different shapes, and the shapes are moving. These pockets or hotspots that some older programs target are non-existent, and they are in fact fictional. And so SILCS is able to find the real hotspots and identify ones that will be targetable by drugs. So in addition to finding these hotspots, SILCS is uniquely qualified to show how to link these hotspots and sort of grab them all at once, which is again a very important component of drug design.
Now, in this representation, we've taken the information from the molecular dynamics simulation that we just looked at, and we've calculated the three-dimensional probability distributions or fragment maps. And here I'm showing the fragment maps for benzene in purple. And what one can see is the locations on the surface of the protein to which benzene wants to interact. We can expand that to now also include the fragment maps for propane, as well as for hydrogen bond donors and hydrogen bond acceptors. And we can see now how we can differentiate the locations of different types of functional groups and where they want to be located on the protein surface. This is the power of the SILCS technology. We can use this information for any ligand, for any position on the protein surface. For example, I'll now bring in the structure of a ligand, which I show here. And what one sees when we compare these structures is we've got the ligand that's bound to the protein, we see the structure of the ligand, and we see the fragment maps actually overlapping the ligand. This shows how the fragment maps can predict the binding location of a ligand. We can see fragment maps located adjacent to the ligand. We could expand the structure of this ligand to add a hydrogen bond donor group. We would predict that the binding affinity of this ligand to the protein would increase.
One of the major advantages of the SILCS system is that it creates a framework for developing a new molecule that a pharmaceutical company or biotechnology company can use immediately. I think that this is going to be very appealing to the pharmaceutical industry and to the biotechnology industry because they have a mechanism now where they can really take a look at what's the best molecule considering a particular protein.
By bringing more ligands and drugs through the drug discovery process, this will maximize the potential for success and allow for more rapid commercialization of the product, be it a drug, an herbicide, a pesticide, or whatever the company is interested in.
As you can see, the SILCS technology is fully functional and currently in use at the School of Pharmacy and the School of Medicine. A startup company based around this platform technology, in conjunction with the university, could quickly generate revenue. The university is actively seeking entrepreneurs to lead in these efforts.